physiological responses mediated by phytochromes. The first one is seed germination. We have discussed examples are lettuce seeds. So here red light is supporting germination. Far red light inhibiting germination. Shade avoidance. That means plant avoids shades and bend towards the sunlight. Plant detects the shade by low red or far red ratio. Responses. So it elongates the stem. It reduces branching. It promotes early flowering. Memorize them. The third physiological activity is deetulation. What is deetulation? So plant initially bends towards the sunlight. If it receives sunlight, so it won't bend towards the sunlight. It absorbs the light. So it stops bending towards the sunlight. We are seeing etulated seedling. What is mean of it? Etulation means bending towards the sunlight. Okay. What is the characters of etulated seedling? Long hypocotyle. Okay. This is long hypocotyle. Closed cotyledons. What is its means? Seed contains. They are like leaves. Okay. But they are not real leaves. So we call it as cotyledons. So what is happening here? They are closed. No chlorophyll because there is no sunlight when etulation happening. So there is no chlorophylls. Light causes. That means when there is sunlight, chlorophyll synthesis is happening here. Leaf expansion. That means cotyledons expansion happening. And also inhibition of hypocotyle growth. Bending towards sunlight is stopping. Already it is getting sunlight. Why it has to bend towards sunlight again? So it is stopping bending. Okay. It is before sunlight. It is after sunlight. Okay. The fourth physiological activity is flowering. Phytochromes regulate photoperiodic flowering. It is like a timetable or clock. Okay. Another important photoreceptor is cryptochromes. They are discovered during the studies about blue light. They are flavoproteins related to DNA polymerase enzymes. Okay. These are important keywords. Remember them. Okay. Cryptochromes consists of PHR domain, photolyase homologous region. The functions of it are, so it binds to chromophores and absorbs blue light. The second point is C-terminal extension, that is CCE. It is important for signal transduction protein interactions. Chromophores in cryptochromes. There are two main cofactors. They are FAD, that means flavin adenine dinucleotide. The second one is pterin. These molecules absorb blue light. It is between 450 nanometers. Types of cryptochromes. There are three types of cryptochromes. They are Cry1, Cry2, Cry3. Functions. Cry1 inhibits hypocotyle inhibition. Cry2 photoperiodic flowering. Cry3 chloroplast or mitochondria. So what is Cry1 doing here? In the blue light, hypocotyle elongation is inhibited. That means bending towards sunlight is inhibited. Mechanism of cryptochrome action. Blue light absorption by FAD. So it absorbs the blue light using FAD. Reduction of FAD. So it can also reduce the FAD. Conformational changes. Interaction with signaling proteins. Cryptochromes regulate gene expression by interacting with COP1, that is constitutive photomorphogenic 1. So what is happening here? Cryptochromes regulating the gene expression by using COP1. Okay. Normally COP1 degrades light response transcription factors. So what is COP1 doing here? It is degrading light responsive transcription factors. So it is no longer useful for light responsive factors. So it has to be inhibited, right? Under blue light, cryptochrome inhibits COP1. So when there is blue light, it is inhibiting the COP1. So what happens now? Photomorphogenesis is happening. What is photomorphogenesis? Photomorphogenesis includes germination. Okay, seed germination happens. Deetulation, leaf expansion, and also photoperiodism. It is like internal clock. Okay, so it maintains night light, daylight, reacting towards night light, daylight. Okay, we call it as photoperiodism. Okay, the first function is inhibition of hypocotyle elongation. So what is happening here? It is stopping the hypocotyle elongation. Why? If there is sunlight, there is no need to elongation. Okay, regulation of circadian clock. So maintaining internal clock. Okay, photoperiodic flowering when there is sunlight. Okay, anthocyanin synthesis. Anthocyanin also a pigment. It is useful for survival, like we apply sunscreen to the face, so it protects from the sunlight. It also attracts the pollinators. It is also useful in stress conditions. So what is happening here? Anthocyanin synthesis is happening here. The third receptors are phototropins. Phototropins are blue light receptors responsible for phototropism. What is phototropism? Remember sunflower tree. What it does? It bends towards the sunlight. So it senses sunlight. After it senses sunlight, axons moves toward the shaded area. What are the functions of axons? Did you remember? It elongates the cells. So cell elongation happen in shaded area. That means this side. What happens if there is more elongation? It bends towards right side, right? So it can receive sunlight. We call this process as phototropism. Remember sunflower tree. Phototropin. There are two major types of phototropins. They are FAT1, FAT2. The function of it is it works in low light responses. FAT2 works in high light responses. These FAT1, FAT2 are responsible for chloroplast movement, stomatal opening, rapid inhibition of stem growth. Now see the diagram. Here is low light, here is high light. Okay, if there is low light, what is happening here? Here is low light, so that means there is blue light. So chloroplast have actin site. So CHUP1, that means CHUP1 binds with the chloroplast. So it pulls the chloroplast towards the shaded area or low light area. Why it is happening? Because if there is high light, then chloroplast will be degraded. So it has to be protected. So what is happening here? FAT1, FAT2 are sensing the high light. So they bind with the chloroplast actin protein that present in the chloroplast. So this CHUP protein binds there and it pulls the chloroplast towards this side. So it avoiding the highlight and protecting the chloroplast. This is the overall idea of it. We can call it as avoiding highlight. Phototropins contain love domains. They are love 1, love 2. Love means light oxygen voltage. ये प्यार नहीं है. These bind FMN. They bind to FMN. Okay. The second domain is serine threonine kinase domain, responsible for signal transduction. Mechanism of phototropin action. What is the mechanism? Blue light activates love domain. Conformational changes occur. Conformation changes means changes in the protein structures. Okay. The third one is autophosphorylation of kinase domain. So phosphorylation is happening here. Activation of downstream signaling. Remember them, I cannot explain you. Phototropin mediated responses. Phototropism means growing towards the light. The second function is stomatal opening. The third function is chloroplast movement. We have seen it. The fourth function is leaf positioning. We have not seen it. Let us see how stomatal movements happen. You can see this stomata is opened. So air and water. Water means transpiration. Both functions happen if the stomata is opened. Here stomata is closed. That means no air is going, no transpiration is happening. These are guard cells. I explain you how this process is happening. In normal situations, that means when stomata is open situations, there is more protons in the guard cells. Protons means hydrogen ions. There are more hydrogen ions in it. So what is this means? There is more acidic and there is more water. Okay. When these guard cells receives blue light, okay, when they observe blue light, proton ATPase activity increases. Okay, so these protons export to outside, that means go to outside. So here is proton loss is happened. Due to that, potassium ions enter into it. So there will be more potassium ions. That means there will be more basic. So in order to cover this basicity, water has to move towards the stomata. That means to guard cells. When the water goes towards the guard cells, they expand and opens the stomata. So this is the process. I can tell you again, the first thing that is happening is when blue light is there, proton ATPase is activated so that protons which are present in the guard cells export towards outside. So now potassium molecules enter into the guard cells. In order to balance this imbalance, now water enter into the guard cells. So what happens now? Turgidity increases. What is turgidity? Pressure will be rises. So it will be extend like sea. Okay, like kidneys. What happened now? Stomata is opened. So now air can pass and also transpiration will be happen. So this is the overall idea.